So what we're going to do in this lecture is examine the individual muscle fibers. Okay? So again, if we look at a whole muscle here, like the biceps muscle, and then we look inside, we'll find fascicles. Inside of those fascicles, here's one that's been extracted, right? That's an F, that's a fascicle. Inside of that fascicle, there are individual muscle fibers. This is one individual muscle fiber that's been pulled out of a fascicle, also called a muscle cell. Let's go ahead and take a closer look at this muscle fiber. Here is one muscle fiber. Okay. What we want to do is take a look at what a muscle fiber is ultimately made up of. Since a muscle fiber is in fact a cell, there's a couple things right off of the bat I want to point out. It has to have a plasma membrane. Every single muscle fiber is a cell, so it must have a plasma membrane. In the case of a muscle fiber, the plasma membrane is called the sarcolemma, but the sarcolemma is just the plasma membrane. It's just named after the muscle fiber. Sarco always is implying muscle. Okay? So a sarcolemma is the plasma membrane of a muscle fiber. What also does a cell have to have? It has to have a nucleus. Okay? Muscle fibers are actually multinucleated, which is beyond the point right now, but every muscle fiber has a nucleus. Okay? So every muscle fiber has a plasma membrane, which is called the sarcolemma. Every muscle fiber has a nucleus, and then inside of the muscle fiber, you find what's known as the cytoplasm, right? All the space inside of the fiber that excludes the nucleus that everything exists within. By that, I mean it's where all the organelles float around. So take a brief second to think about what all that means. Extract, okay, you say the biceps muscle has, let's say, 300,000 muscle fibers. Extract any one of those fibers, and any one of those 300,000 muscle fibers will have a plasma membrane, it will have a nucleus, and it will have a cytoplasm. It has everything that is required for something to be called a cell. So every muscle fiber is a cell. That's why you can call them muscle cells. And this is where, things get a little bit different, because we have trillions of cells in the human body, but a lot of those cells are very different from one another, right? They all have a plasma membrane, a nucleus, and a cytoplasm in order to be a cell. But then when you start getting beyond that, you begin to see differences between the cells for obvious reasons, right? We need different cells doing different things. So what makes a muscle fiber a distinct type of cell that we call a muscle fiber? And the answer is this very unique organelle that it has inside of it. If you look inside of a muscle fiber, okay, you find all of these cylinder-shaped structures inside of the fiber. They're actually an organelle that's called a myofibril. Okay? Each one of these cylinder-shaped structures inside of this muscle fiber, this muscle cell, is called a myofibril. Myofibrils are organelles. And myofibrils, if you look inside of a muscle fiber, this specific organelle that's called a myofibril makes up like 80 to 90% of the actual muscle fiber itself. Most other cells in the human body don't have myofibrils, these cylinder-shaped structures, and they certainly don't take up, most other cells in the body don't have myofibrils, and those myofibrils definitely don't take up like 80 to 90% of the cytoplasm, right? The space inside of the cell that doesn't include the nucleus. If you look at a muscle fiber, specifically, we're just to remind you, we're focusing on skeletal muscle right now. So if you take a look at a skeletal muscle fiber, a muscle fiber inside of a whole skeletal muscle, you'll find out that 80 to 90% of its cytoplasm is taken up by these cylinder-shaped structures called myofibrils. And that's actually what makes muscle fibers, a unique and different type of cell. It's really the organelle, the myofibrils that exist within them. That's what's going to make a skeletal muscle fiber different than a neuron, for example, right? A neuron and a muscle fiber are both cells. They both have a plasma membrane, a nucleus, and a cytoplasm. But, what makes the skeletal muscle fiber different than the neuron, right, is the fact that the skeletal muscle fiber has all these really unique, it's one type of organelle, but it's packed with this unique type of organelle that we call a myofibril. So if that's what makes a skeletal muscle fiber unique, right, different than other cells in the body, then clearly we need to take a closer look at the way that these myofibrils are organized. So here's an image of a portion of a myofibril. Okay, right now we're just focusing on this organelle that exists within skeletal muscle fibers called a myofibril, this cylinder-shaped structure. And one has been extracted here. That's only actually a portion of one. All right, so let's say this. We'll get into more detail in a second, but this is a good point to start with. Myofibrils are made up of repeating segments of what are called sarcomeres. Another way of saying that is myofibrils are made up of sarcomeres. They're made up of sarcomeres that just repeat over and over and over and over and over again down the length of the myofibril. What I always say is myofibrils are made up of repeating segments of sarcomeres. That is what a myofibril is ultimately made up of. And I keep compartmentalizing all of this for a reason. And that's because if you say myofibrils are made up of repeating segments of sarcomeres, then if you want to understand a myofibril and thus understand a muscle fiber, since muscle fibers are made up of myofibrils, then you better take a close look at the actual sarcomere itself, right? Because that's really going to be the functional unit of the skeletal muscle fibers. It's the sarcomeres that make up the myofibrils. That's where you're going to find these contractile filaments within skeletal muscle fibers that actually generate force, right? You can go out to the broadest picture possible, and you can say the purpose of a skeletal muscle fiber is to contract and generate force. We did that in the first lecture, okay? But then when you really focus in on the skeletal muscle fiber, and you say, well, how does it generate force? The first thing you have to recognize is the muscle fiber is made up of these myofibrils, these cylinder-shaped structures inside. And the cylinder-shaped structures, the myofibrils, are made up of these repeating segments of sarcomeres. And then if you look at the actual sarcomeres themselves that make up the myofibrils, you can finally understand how the skeletal muscle fiber actually contracts. It generates force because force production is going to occur within these things that we call sarcomeres within the myofibrils. So let's take a look at one sarcomere within a myofibril. So what does a sarcomere look like? First of all, let's just talk about the length of the sarcomere. The whole sarcomere goes from what's called Z disc to Z disc. Right? Each one of these is a Z disc. Now, the way that I always used to remember this, I'm going to erase this one real quick, but the way that I always used to remember this is because I would say that the Z discs, kind of just look like repeating Z's. But that's not actually, the Z actually comes from a German word that means see-through. But it was always very easy for me to remember Z discs because, well, again, they're in the shape of Z's and it's called a Z disc. Anyways, from Z disc to Z disc is the whole length of the sarcomere. Okay? From Z disc to Z disc, that's the whole length of the sarcomere. So great, you're off to a good start. Next, what also is involved with the sarcomere? And these, now we're starting to get into the filaments that a lot of people have heard of before. The first one is what's called the thin filament actin. The thin filament actin comes off of the Z discs. Okay, each one of these is an actin filament. Every single one of these, right? They're all actin filaments. They're called thin filaments, and they come off of the Z discs. All right, in the center of the sarcomere, the dead center of the sarcomere, there's a protein filament that's in a straight line, and it's called the M line. The M line is this centrally located protein filament within the sarcomere. Okay, it's the dead middle of the sarcomere. All right, now what comes off of the M line? What comes off of the M line is what's called the thick filament myosin. The thick filament myosin is attached to and comes off of the M line, and as it does, it overlaps to some degree the actin filaments on both sides. Not completely overlaps, but does partially overlap the actin filaments on both sides. So again, the thick filament myosin attaches to the M line and slightly overlaps the actin filaments on both sides. So each one of these is a thick myosin filament. All right. Now there's much more to talking about the sarcomere than this, because there's some little intricate proteins that we need to discuss. But on a basic level, this is the sarcomere. Like this is how you would learn about the sarcomere, in a very, let's say introductory, but also relatively complete way. A sarcomere is made up of two Z discs, first of all, right? And from one Z disc to the next is the length of the sarcomere. And then the thin filaments, actin, come off of the Z disc and head towards the center of the sarcomere. Then in the direct middle of the sarcomere, you find the M line. And what comes off of the M line is the thick filament myosin. And as it does, it slightly overlaps actin on both sides. And there you have it. Like that's one sarcomere. And these protein filaments, actin and myosin, within these sarcomeres are what ultimately are going to allow for force to be produced during a contraction within the muscle fiber. And we're going to, progress and get to that very shortly here. But for now, I just wanted to show you the basics of one sarcomere. And the next most important thing to realize here is within a myofibril, you find a sarcomere, but the sarcomeres just repeat over and over and over and over and over again down the length of the myofibril. This structure will repeat again and again and again and again down the length of the myofibril. And let's try to take a look at that. So yet again, we're looking at this myofibril. Okay? And within this myofibril, we find sarcomeres. Right? What makes up a sarcomere? Well, first of all, the length of the sarcomere goes from Z disc to Z disc. Right? So there's one sarcomere right there. Right? And then what comes off of the Z disc? Well, the thin filaments, actin. Well, let's draw them in a different color. How about we do them in red? The thin filaments, actin, come off of the Z disc. I'm drawing them in red. Okay. In the center of the sarcomere, there's the M line. Okay, there you can see the M line. And what comes off of the M line is the thick filaments, myosin, which we can draw, how about, in blue. And you can see that those myosin filaments slightly overlap actin on both sides of the sarcomere. Okay, so there you have it, right? If you look at the myofibril, you're now visualizing one sarcomere. And the best part is, right, it's these sarcomeres and thus myofibrils are relatively easy to understand because the sarcomere just keeps repeating over and over and over again. I mean, look to the right, right? There's another sarcomere, right? Z disc to Z disc is the sarcomere. There's the actin filaments coming off of the Z disc in the center. There's the M line, and then the thick filaments myosin are coming off of the M line and overlapping actin, right? So there's another sarcomere right next to this sarcomere. You go to the right again, right? There's another sarcomere. And if you were to keep going, it would just be another sarcomere, another sarcomere, another sarcomere. The sarcomeres would just keep repeating down the length of the entire myofibril. And that'd be true on the left side over here too, right? There would just be another sarcomere, another sarcomere. That is what a myofibril is made up of. A myofibril is made up of these sarcomeres that just keep repeating again and again and again and again and again and again down the length of the myofibril. So again, we don't want to lose sight of what we're trying to understand, which is always easy to do when you start getting into more detailed physiology or detailed anything. You can lose sight of the purpose of everything. And the purpose of all this is ultimately to understand how a muscle fiber generates force, because we know where that force is relayed onto and what it results in, right? Movement of the skeleton. So that's what we're trying to do here. And what we're doing is we're looking at this one muscle fiber, right? And recognizing that the muscle fiber is made up of myofibrils for the most part, right? That's the most common organelle. It's packed with this organelle that is referred to as a myofibril. It's this cylinder-shaped structure. And this cylinder-shaped structure that's called a myofibril is just made up of repeating segments of sarcomeres. Right there you can see it. You can see this image, right, that we've been looking at for a very long time now in these different lectures. You can see one myofibril being pulled out of a muscle fiber. And you can see even in this image that the myofibril is made up of these repeating segments of sarcomeres. And now you have an idea of what the sarcomere looks like. And I've told you that within the sarcomere, those actin and myosin filaments are where force is actually produced. So at the level of the sarcomeres within the myofibrils is where force production happens within a muscle fiber. So clearly what we need to do now is focus in even more so on the actin and myosin filaments of the sarcomeres. We know how the actin and myosin filaments are organized within the sarcomeres of the myofibrils, but we need to focus in on the actin and myosin filaments to actually understand how they interact to generate force. All right, so here is the muscle fiber. And this is one myofibril being pulled out of the muscle fiber. We know that the myofibrils are made up of sarcomeres, right, from Z disc to Z disc. And we know that within the sarcomeres, there's mostly the thin filament actin and the thick filament myosin. Okay, that's where the interaction between the actin and myosin filaments within the sarcomeres of the myofibrils is where force is going to be produced. So we need to focus in on these protein filaments, and we're going to start off with actin. So at the bottom here is what the actual actin filament looks like that comes off of the Z disc. Okay, if that's a Z disc right there, this would be the actual actin filament that comes off of it, and that's what it kind of looks like. Okay, so there's a lot that we need to discuss before we can, finally see and understand what this true actin filament looks like. So first of all, what's the actin filament actually made up of? And it's made up of all these little globular proteins that are called G-actin. G stands for globular. Okay? All those little globular proteins called G-actins, what they do is they actually come together to form a little strand that's called F-actin. F-actin is made up of a whole bunch of G-actins, little globular actins, globular proteins that all come together to form a little strand. Okay, we're all going to ultimately get to the point of understanding the true actin filament. Okay, so all these globular actins, these globular proteins, come together to form a little strand that we call F-actin. And then the next thing that you need to do is you need to take two F-actin strands and wind them around each other in a double helix, okay? Like that. If you take two F-actin strands and you wrap them around each other in a double helix like DNA, then you will get the major part of the actual actin filament. The actin filament itself that is actually attached to the Z disc is made up of two F-actin strands wound around each other in a double helix. Okay. Now, as you can clearly see, there are some other proteins located on the actin filament that we absolutely need to discuss. Okay, so we're not done talking about actin yet, but the majority of the actin filament attached to the Z disc is actually made up of two F-actin strands wound around each other in a double helix. And of course, there's also one other thing that is really important that I didn't mention right off of the bat. If you look at each little globular protein called G-actin, right, globular actin, each one of them has a little black, let's change the color. It has a little black site on it, but I'll color it in red. Okay. Each one of those little black sites, dots, located on each one of these globular actins is what's called a myosin binding site. Okay? And you can see that, right? If you take all these globular actins, these globular proteins, you combine them together to form a little strand that we know as F-actin, and then you take two F-actins, you wind them around each other in a double helix, you can look at the actual actin filament itself that's attached to the Z disc, and you can still see that each one of these G-actins, these globular proteins, still has, every single one of them still has those little black dots on them, which are these myosin binding sites. Now, that's not relevant just yet, but it will be very, very shortly. Clearly, each one of those, if it's called a myosin binding site, that's where that thick filament myosin has the ability to bind to and interact with actin. So just keep that in the back of your mind for now. And ultimately, it's actually relevant for what we're about to talk about in a second here. But it's not all going to come together for a few more minutes. These myosin binding sites on every single one of these globular actins, these G-actins, is clearly the location, ultimately, where the thick filament myosin can interact with actin. And remember, the interaction between these two proteins, as I've said many times, is that's where force is actually produced. Before we get to force production, though, we need to recognize that the actin filament has some other proteins that are associated with it. We have to talk about those. So here is the actin filament again. Okay, this is what would actually be attached to the Z disc. All right. It's made up of two F-actin strands wound around each other in a double helix. Now, look at this. There is this little strand-like protein that runs along the actin filaments, right? It actually runs along the two F-actin strands. And what this little protein does, first of all, its name is tropomyosin. This little strand-like protein that runs along the F-actin strands of the actin filament is called tropomyosin. And if you look closely, it runs along the F-actin strands in a perfect way, in so that it covers up the myosin binding sites on every single one of these globular actins, these G-actins that actually make up the entire actin filament itself, right? And tropomyosin covers up the myosin binding sites on all of the different G-actins and all the different globular actins that are making up the F-actin strands that are wound around each other, creating the actin filament. So I don't think actually need to get to the next part of this to understand what this means. It means as long as this strand-like protein called tropomyosin is covering up these myosin binding sites on all these different, globular G-actins, as long as tropomyosin is there, myosin, the thick filament, can't actually interact with actin at all. It would be impossible to, because clearly the myosin binding sites on the different globular actins, the G-actins, is where myosin interacts with actin. And as long as tropomyosin is covering it up, how's that going to happen? And the answer is there's another protein that we have to take a look at called troponin. So we have this troponin protein. Troponin, it's called the troponin complex actually because it's made up of a variety of different proteins. The different proteins within troponin, we're not going to get into the names of all of them, but I will tell you what they do. Okay, the troponin protein attaches to both actin, right? And to be clear, real quick, okay, the actin filament itself, if I haven't been clear about this, the actin filament itself is made up of two F-actin strands wound around each other in a double helix. That's what makes up the actin filament itself. Tropomyosin is associated and plays a very important role in regulating actin, but tropomyosin is not technically, you might hear people disagree with this, but tropomyosin is really not technically part of actin. Okay, tropomyosin is associated with actin, and it clearly plays a role in regulating actin by covering up the myosin binding sites on the different G-actins that make up the actin filament. But tropomyosin is a different protein than the actin filament itself. Okay, and I say that for a very specific reason, because as I talk about troponin, troponin binds to both actin, right, the actin filament, and it binds to tropomyosin. Okay, because they're really two different proteins, right? The actin filament and tropomyosin are different proteins, and the troponin complex, this series of proteins, binds to both of them. And what can happen is troponin has a binding site on it for calcium. And when calcium binds to troponin, it causes a conformational shift in the troponin protein. And that conformational shift in the troponin protein, what it does is it pushes tropomyosin off of the myosin binding sites on the different globular G-actins. This is going to become incredibly important, right? If you can get calcium to elevate, calcium ions to elevate inside of a muscle cell, then what calcium has the ability to do is calcium has the ability to bind to troponin. And when calcium binds to troponin, what that does is it causes a shift in the troponin protein. And that shift in the troponin protein pushes the tropomyosin strand, which troponin binds to, right? Troponin binds to tropomyosin. So if there is a shift in the troponin protein, it can push the tropomyosin strands off of the myosin binding sites on actin, right, on the globular actins. And this, in theory, would free up the ability of the thick myosin filaments to now be able to interact with actin. So for the sake of this class, this is the extent to which you really need to know about actin. That's the extent to which you need to know about actin, right? Here is the actin filament, right? In a normal state, the actin filament has these strand-like proteins on it called tropomyosin that cover up all of the myosin binding sites on the globular actins within the, that make up the actin filament. Okay. But there's also this protein complex called troponin that binds to both actin and tropomyosin. And if calcium can bind to troponin, that will cause a shift in the conformational structure of troponin, and that will push the strand-like proteins, tropomyosin, off of the myosin binding sites on the globular actins within actin. So this is yet another rendering of a sarcomere, and you are seeing the actin filaments come off of the Z discs. Now, the reason that I'm using this image of a sarcomere compared to the ones I was using previously is because this image is a little bit more detailed and it looks a lot more like the images of actin that I just showed you. Clearly, tropomyosin and troponin are missing from this. You can, I hope, imagine what those would look like, but overwhelmingly, this image is much more detailed than the prior ones. So hopefully after seeing those images and descriptions of actin, tropomyosin, and troponin, seeing this this image of a more detailed sarcomere is even more helpful. So what we need to do now is focus in on the thick filament myosin that comes off of the M line and overlaps actin slightly on both sides. This image, none of the images of a sarcomere are ever perfect, which is why I use so many different images of it. This one, to begin with, the myosin and actin filaments are far too overlapped here compared to what they would really be. You wouldn't have this extreme of overlap, certainly in a resting circumstance. But nonetheless, it's still a very detailed image of a sarcomere, and we're going to keep using it. And the myosin filament that we're about to look at looks very similar to the way that myosin is depicted in this schematic. So that's my rationale for using this. Now, on first glance, it looks like the myosin filament, the thick myosin filament that comes off the M line, has all these little heads that come off of it, right, called myosin heads. Right? And it almost looks like in the image that the myosin heads are like this other protein that are associated with the myosin filament, and it's really not like that at all, which is why I hadn't introduced or haven't introduced the myosin heads up until this point. Because when you do this on an introductory level, what you'll say is that the thick filament myosin comes off of the M line, and then what's a part of the myosin filament are the myosin heads. Okay, but that's pretty difficult to say and make sense of because it kind of makes the myosin filament sound like it's different from the myosin heads, and it's really not. Okay, what this myosin filament is made up of are myosin proteins. Okay, if you think of the actin filament, what is the actin filament really made up of? It's made up of a whole bunch of, globular actin proteins. The actin filament is made up of these, globular actins, which are, this spherical type of protein. Okay? The myosin filament is made up of a whole bunch of myosin proteins. Okay? And the heads are actually a part of those proteins. Let me try to explain. Is a myosin filament, right? What the myosin filament is actually made up of, again, are a whole bunch of myosin proteins, which are right here. Okay. Now, the myosin proteins, in reality, it takes a different course to get into the whole explanation of a myosin protein, but we can go through the basics of it. Okay, so what a myosin protein has, first of all, it has a tail, and then from the tail, there are two heads that are formed. Okay, that's what a myosin protein is made up of. It's made up of a tail, and it's made up of two heads. Okay. You can see it here too, right? There's a tail, And those are the two heads of the myosin protein. And how are the heads organized, first of all? Okay, let's go back down here. Okay. This is a whole myosin filament. Okay, very important, filament. And the myosin filament, right, this structure here, is made up of a whole bunch of myosin proteins, which are up here. The myosin proteins have a tail, and they have two heads. Now, the two heads are orientated so that they are angled in a way that they actually face the actin filament. Okay, so let's say it this way. Let's try to use some better language. The tails of the myosin proteins inside of the myosin filaments. Right? You can actually see the tails if you look really closely. All the tails are in here. Okay? The tails, let's say, are parallel to actin. Right? If you look at the myosin filament, the tails of the myosin proteins in the myosin filament are parallel to actin. But the heads, because remember, a myosin protein is made up of a tail, and then there are two heads. The heads of the myosin proteins are more so perpendicular to actin, meaning that they're angled in a way that they actually face actin. Let's look at a more blown up image of this. Here you can see the whole myosin filament. Okay? Inside of the myosin filament, there are those myosin proteins, and you can actually see, if you look closely, there you can see the tails. Okay? So the myosin proteins have tails, and the tails are parallel to actin. Now, the protein has a tail, and it also has two heads, and the heads are orientated and angled in a way so that the heads lie more perpendicular to actin. The heads actually face actin. And that makes more sense when you look at what exists within or on the heads. So within each head of a myosin protein that exists within the myosin filament, within each head, there's first of all a site that binds and breaks down ATP. It's called an ATPase site. And additionally, within each head, there's what is called an actin binding site. Well, that's interesting, right? Kind of the tip of each myosin head, at least that's the way that it's rendered in images, has what's known as an actin binding site. Well, that's interesting. That must imply that the heads, then, of the myosin proteins are capable of binding to actin. Which brings us back to the question of, well, then where exactly do the myosin heads bind onto actin? We're minutes away from discussing the cross-bridge cycle, but we're not quite to that point. I'm using this image for a very specific reason. Okay. First of all, here we would see the kind of myosin, a portion of the myosin filament. What's within the myosin filament? The myosin proteins. Okay, here's one myosin protein, right? There you can see its tail, and there you can see its two heads. All right. Within the two heads, there are actin binding sites. They would be like here. That must mean that those actin binding sites are capable, the actin binding sites within the myosin heads, are capable of binding onto actin. And then you say, well, where do the myosin heads, the actin binding sites on the myosin heads, bind onto actin? And the answer is, well, if we look at actin, right, what's actin made up of? It's made up of a whole bunch of globular actin, these G-actins. And what do the G-actins have on them? Myosin binding sites, right? All those little black dots there, I'm drawing in red, on the different globular actins, those are myosin binding sites. So without getting it, let's skip some of all the terminology and the name calling because it just makes it more confusing. clearly what's going to happen here? The myosin heads are going to latch on to actin. Specifically, the myosin heads are going to bind on to the globular actins, specifically the myosin binding sites within the globular actins. And that is how myosin and actin actually interact. Right? Myosin and actin interact by the myosin heads of the myosin proteins grabbing onto the different globular actins within the actin filament. And then the question becomes, well, how do they two actually interact in a process, which we'll talk about in just a second here. I just want to try to make the point as to how this structural organization of the sarcomere allows for myosin and actin to interact at all. And hopefully now you can look at a sarcomere and you can understand that. Right here are the Z discs, okay, in black. What comes off of the Z disc? The thin filament actin. Okay. What is actin made up of? You can actually see it in the image, right? Actin is made up of all these different globular actins, these G actins. Okay, that's what makes up the actin filament. Okay. And then in the center of the sarcomere, there's the M line. What comes off of the M line? The thick filament myosin. And what is myosin made up of? It's made up of a whole bunch of myosin proteins. And what are the myosin proteins? You know, how are they organized? Well, they're organized so that the tails within the myosin proteins, you know, that make up the myosin filaments, the tails lie parallel to actin. And the myosin heads are angled so that they're more perpendicular to actin, right? The heads of the myosin proteins. Right? The tail region of the myosin proteins is parallel to actin, and the heads are slightly perpendicular to actin. And then what happens? I mean, because of this organization where the myosin heads are perpendicular to the actin filaments, that allows for the myosin heads to be able to latch onto, look at, it allows for the myosin heads to be able to latch on to the actin filaments. Specifically, the myosin heads will latch on to the different globular actins. Right? The myosin heads will latch on to the globular actins at the level of the myosin binding sites, right? Because each globular actin has, within the actin filament, has a myosin binding site on it. And what that means is the myosin heads are capable of binding to the myosin binding site on the different globular actins. That is how the sarcomere is organized to allow for myosin and actin to actually interact with each other. The myosin heads lie perpendicular to actin, and the actin filaments have binding sites for the myosin heads. So the myosin heads clearly can latch onto and bind to actin. Now you get into a whole different conversation about how the interaction between the myosin heads and actin is accomplished, for one thing, because this is going to be an energy consuming process. And secondly, how does this result in force being produced? Right? Because the interaction of the myosin heads and actin is actually where force production happens. The interaction of the myosin heads with the actin filaments, with the globular actins, that's where force is produced. And that force that's produced at this level is what's actually relayed onto the endomysium, tendon, and thus bone. And that brings us to the cross-bridge cycle. This cross-bridge cycle allows us to actually see how the myosin heads and actin interact. And ultimately, we can use this information to understand how force is produced and then how it gets relayed onto the tendon and bone. So first of all, we kind of have to start in what's this image down here. That's called the rigor or the Riger state. That's where the myosin head is actually actively bound to a actin, globular actin, to the actin filament. The myosin head is actually bound to a myosin binding site on a G-actin. And then what happens is a molecule of ATP comes along and it binds to the myosin head. And we know that when ATP, remember the myosin head has an ATPase site in it, right? It's a site that binds ATP. When ATP binds to the myosin head, what that does is it allows for the myosin head to actually release itself from actin. Do you see in this image down here, the myosin head is actually actively bound to actin? And then when ATP comes along and binds to the myosin head, that allows for the myosin head to release itself from actin. Okay, it actually will begin to cock backwards. Okay, and as the myosin head is moving backwards, what will happen is the ATP will get hydrolyzed by the myosin head. Right, it's the site that's in the myosin head is called an ATPase. That means the myosin head actually has the capacity to break the molecule of ATP down. And the interesting thing is when the ATP molecule gets broken down, it actually stays within the myosin head. Like the ADP and the inorganic phosphate actually stay within the myosin head when ATP is hydrolyzed. So what happens next is once the ATP is hydrolyzed to ADP and inorganic phosphate, it stays within the head, and the head moves back upwards, and it grabs on to a new globular actin. It grabs onto a new myosin binding site on a different G-actin. And then what occurs is the inorganic phosphate is actually released from the myosin head. And this results in the power stroke. What happens with the power stroke is the myosin head actually swings or cocks forward. Right? You can see that from here to here, right? That's one step to the next. Right now, the myosin head is bound to actin, right? The myosin head is bound to a myosin binding site on a G-actin, and inorganic phosphate and ADP are still within the head. And then what happens is the inorganic phosphate is released. And when that inorganic phosphate is released, we think that's when the power stroke happens. And the power stroke is when the myosin head actually swivels forward. And the myosin head, when I say swivels forward, it swivels actually towards the M line. The myosin head swivels towards the M line. And as it swivels towards the M line, what does that do? It pulls the actin filament towards the M line. Right? Every single time the myosin head swivels forward, it's always going to be pulling the actin filament towards the M line. Okay? And from the power stroke, like when the myosin head swivels forward and it's latched on to actin, from the power stroke, actin actually moves towards the M line. And then in the last step, ADP is released, and you have returned to what we started with, which is this rigor or Riger state. Depending upon who you speak to, the term is different. Okay? All right. So now you're back to exactly where you started, right? The myosin head is bound to actin, right? And it's just stuck there. It's bound to a myosin binding site on a G-actin. So what has to happen next? Another molecule of ATP has to come in, bind to the myosin head, and that will allow the myosin head to release itself from actin, and it'll cock backwards. And as it does, the myosin head will consume the ATP, right, hydrolyze it, and it will turn into ADP and inorganic phosphate. And as that happens, the myosin head will move upwards and grab on to a new G-actin, right? A myosin binding site on a new G-actin within the actin filament. And then you'll see the power stroke again. So the inorganic phosphate, will be released, and the myosin head will swivel forward again. And what will that do? If the myosin head swivels forward, it will pull the actin filament yet again closer towards the M line. And this just keeps happening during a standard contraction. This, as you'll see at the end of the lecture, at least the next lecture, this sort of motion doesn't always go on endlessly or sometimes at all, but this is the natural direction that the actin filament is pushed towards during your typical contraction, what's called a concentric contraction. We'll get to that. This is what everybody thinks of. Okay, the myosin head is constantly cycling in a direction that pushes or pulls the actin filament towards the M line. So we can look at this image and we can tell what's going to happen, right? If these myosin heads are cycling, right, swiveling, what are they doing? The myosin heads, they're first of all angled in a way that they can interact with the actin filament, right? The myosin heads that are part of the myosin protein are angled so that they're slightly perpendicular to the actin filaments. And then they're constantly swiveling or cycling so that they pull the actin filament towards the M line. That is what the myosin heads do. Their natural ability and function is to grab on to the actin filaments, right? They're obviously grabbing on to different G-actins, myosin binding sites on different G-actins, and swiveling and thus pulling the actin filaments towards the M line. That's what happens during a contraction. During a contraction, the myosin heads cycle, and as they cycle, they continuously grab onto different sections of actin, by that I mean different G-actins, and pull those G-actins and thus actin filaments towards the M line of the sarcomere. Now this is a much less detailed image of what we've been talking about, but it still drives home the major point. Here you can see a sarcomere. Z-disc to Z-disc is a sarcomere. Now, during a contraction, what's going to happen? The myosin heads are going to cycle, right? They're going to swivel. And as they swivel, they're going to keep pulling actin towards the M line, right? Every myosin head is just going to keep cycling and continuously pulling actin, the actin filament as a whole, towards the M line. The myosin heads are doing that by grabbing onto these different G-actins and pulling each one of them towards the M line. And what's ultimately going to happen to the sarcomere? The sarcomere is going to shorten, right? It's going to get smaller. Why? Because you're going to get more and more overlap of the myosin and actin filaments, right? It only makes sense. If the myosin heads are cycling and continuously pulling actin towards the M line, then more and more actin is going to begin to overlap myosin. You can see that from this top image to the bottom image, right? From the top image to the bottom image, the myosin and actin filaments are overlapping much more, right, than they are on the top image. And what we say about this ultimately is if the myosin and actin filaments are overlapping more, then, of course, the sarcomeres have shortened. That's what generally happens during a contraction. I have to say generally because there are different types of contractions that we will talk about that don't always involve this exact thing happening. But this is what most people think about when they think about a skeletal muscle contraction. During a standard skeletal muscle contraction, the sarcomeres within the myofibrils actually shorten. And it's because of the fact that the myosin heads are continuously pulling the actin filaments towards the M line and thus shortening the sarcomeres themselves. So I'm just going to kind of make a broader point that I think everybody can understand. Okay? If during a contraction, right, inside of a skeletal muscle fiber, what a contraction actually involves is the myosin heads grabbing onto actin and pulling actin towards the M line. Okay. What that's going to result in is the sarcomeres shortening, right? All of the sarcomeres within a myofibril during a contraction are going to shorten. Right? And then you think, well, what does that mean for the actual myofibril itself, right? If all of the sarcomeres within a myofibril shorten during a skeletal muscle fiber contraction, what does that mean for the actual myofibril? Well, the actual myofibril itself will shorten. And then you can blow that out and think about it on a bigger level too. So here is a whole muscle fiber, right? What's a muscle fiber made up of? Mostly, right? The most common organelle within a muscle fiber is a myofibril. Okay, so here's one myofibril being pulled out. What's the myofibril made up of? The myofibril is made up of sarcomeres. If during a contraction, all of the sarcomeres within a myofibril shorten, then what happens to the myofibril? The myofibril shortens because what's the myofibril made up of? It's made up of sarcomeres, and if all the sarcomeres are shortening within a myofibril, then the myofibril itself is shortening. Okay, if during a contraction, all of the myofibrils are shortening because all of their sarcomeres are shortening, what does that mean for the actual muscle fiber itself? And the answer is the muscle fiber will shorten, right? Because what is the muscle fiber made up of? The muscle fiber is made up of like 80 to 90% myofibrils. And if all the myofibrils are shortening because all of their sarcomeres are shortening, then that means the muscle fiber itself must be shortening, right? I mean, again, if the muscle fiber is made up of like 80 to 90% myofibrils, and during a contraction, all the myofibrils are shortening, then of course the muscle fiber itself has to be shortening. And then you can finally take this to its logical conclusion. If a whole muscle, like the biceps muscle, is made up of hundreds of thousands of muscle fibers, okay? The biceps muscle is made up of hundreds of thousands of muscle fibers. What happens during a contraction? Not necessarily all of them, are going to participate in the contraction. Actually, that never happens. You're never going to get every single muscle fiber in a whole muscle to participate in the contraction. But during a contraction of the biceps muscle, many muscle fibers within the biceps muscle are going to contract. And what happens when they contract? Many muscle fibers within the biceps muscle are going to contract when the biceps muscle contracts, because the biceps muscle is made up of muscle fibers. So if many muscle fibers contract within the biceps muscle, what does that actually mean about the muscle fibers? It means that those muscle fibers are shortening, right? Let's say 100,000 out of 300,000 muscle fibers within the biceps muscle are actually participating in the contraction. You'll see why that makes sense later on in these lectures. Okay? If 100,000 of those muscle fibers are contracting within the biceps muscle, then 100,000 of those muscle fibers are shortening, right? Because the myofibrils within those muscle fibers are shortening because the sarcomeres within those myofibrils are shortening. Right? So what does that mean for the whole biceps muscle? Well, if during a contraction, a large majority of the muscle fibers within the biceps muscle contract, and that actually means a large majority of the muscle fibers within the biceps muscle fibers shorten, because that's generally what happens during a contraction. What does that mean for the whole biceps muscle? Well, if the whole biceps muscle is made up of mostly muscle fibers, and during a contraction, many of those muscle fibers are contracting and thus shortening for all the reasons that we've discussed, then the whole biceps muscle itself must shorten. Because if the muscle fibers are shortening and the muscle fibers make up the biceps muscle, then the biceps muscle itself must be shortening. That's the answer, right? The biceps muscle is made up of muscle fibers, and during a contraction, many of the muscle fibers are contracting, and when they contract, they shorten. So if many of the muscle fibers within the biceps muscle, which make up the biceps muscle, are shortening, then the whole biceps muscle must be shortening. And let's talk about what that looks like. So let's think about this. When I want to contract my biceps muscle, what happens? Muscle fibers within my biceps muscle contract, right? Logically, that has to be true. Muscle fibers within my biceps muscle are going to contract. And what does that actually mean? It means many of my biceps muscle fibers are going to contract, and thus they're going to shorten for all the reasons that we discussed, right? Because the muscle fibers are made up of myofibrils, which are shortening because the myofibril sarcomeres are shortening. So many of the muscle fibers within my biceps muscle are going to contract, and thus they are going to shorten during a contraction. And if many of the muscle fibers contract and thus shorten, then my whole biceps muscle must contract and thus shorten. And you can visually see that, right? I mean, what's the biceps muscle doing right now? It's shortening, it's balling up. That's exactly what happens during a contraction like that. Right? The actual whole biceps muscle shortens. You can do an experiment like I just did with yourself. Contract your biceps muscle, and you'll see that the whole muscle shortens. And that's because of the fact that all the muscle fibers, not all of them, but most of the muscle fibers within that biceps muscle, many of them, are contracting and thus shortening. They're all shortening, but they're not all participating in the contraction. You'll see. And then finally, we can conclude with this. Right? When the whole biceps muscle shortens because the muscle fibers within it are shortening, that results in the insertion, the radius, being pulled towards the origin, the scapula. Right? It results when the biceps muscle contracts and thus shortens. What it ultimately accomplishes is it pulls the insertion, the movable bone, the radius, towards the origin, the immovable bone, the scapula. And what do we produce? Elbow flexion. Right? So there's the whole thing. Now, this still didn't address how force gets relayed onto the endomysium and then onto the tendon, onto the bone. Okay? Force is generated when the myosin head and the actin filament, the G-actin, globular actin, interact with each other. The swiveling of the myosin head pulling on actin is actually where force is generated. That's where force comes from. It's through the interaction of myosin and actin. But how does this force that's generated between the myosin head and actin actually ultimately then get relayed onto the endomysium? And most of this is really outside of the scope of this class. You should take a more advanced physiology course if this is something that truly interests you. But I'll explain it in the most basic way possible. Okay, so when the myosin head latches onto actin and swivels, it generates force. And what happens is that force is really generated onto the actin filament. And what's the actin filament attached to? The actin filament is attached to the Z disc. So the point is, if the myosin head interacts with actin and it generates force by interacting with actin, then the force is actually relayed from the actin filament onto the Z disc. And then what you see, there are protein filaments. They're called intermediate filaments. And they actually latch onto the Z disc and they run up and attach to the plasma membrane. So there are these little protein filaments called intermediate filaments. The most common one is named desmin. And it actually runs from the Z disc. It's like a string. And it runs from the Z disc up to the plasma membrane of the muscle fiber called the endomysium. And the intermediate filaments are attached to these things called costomeres, and ultimately those are attached then to the endomysium, which actually lies on top of the plasma membrane. Okay, so the point is basically, because again, we don't have the time to go into all the detail of this, these intermediate filaments that are attached to the Z disc are ultimately attached to the endomysium, not directly, but indirectly. Okay, so here's what happens. During a contraction, the myosin heads cycle, right? The myosin heads grab onto actin, they pull on actin, and that generates force onto actin. And the force is relayed from actin onto the Z disc because that's what actin is attached to. So the force gets relayed from the myosin head onto the actin filament, and from the actin filament onto the Z disc. And then what attaches to the Z disc are these things called intermediate filaments, these little stringy proteins. So the force gets relayed from the Z disc onto the intermediate filaments, and the force gets relayed from the intermediate filaments ultimately onto the endomysium. And then if you follow it downstream, the endomysium becomes the tendon, which attaches to the bone. That is how force, we think, there's a whole field of study that looks into how all this is happening. We're kind of in our infancy in understanding how all this works. But that is how we think the force that's generated through the interaction of myosin and actin within the sarcomeres ultimately gets relayed onto the endomysium and then onto the tendon on the bone. It's because the force gets relayed onto actin, which gets relayed onto the Z disc, which gets relayed onto the intermediate filament, which ultimately then relays the force onto the endomysium. If this kind of stuff interests you, I'll say it again, you need to take more advanced muscle physiology courses because there's much, much more information that we know than what I've just given you here. But this is the basics as to how we think that force that's generated by the sarcomeres ultimately reaches the endomysium and reaches the bone and moves the skeleton. So hopefully I've answered at least a decent number of questions or shown you a couple new things in this lecture. But there's also plenty of things that we haven't answered yet. Okay? And they mostly involve what actually stimulates this contraction. Okay? And we can begin that conversation by saying, what are the two things that need to be elevated inside of a skeletal muscle fiber in order for it to contract? And there's two obvious things. You need calcium and you need ATP. Those are the two things that are 100% required in order for a skeletal muscle fiber, a skeletal muscle cell to contract. Why do you need the calcium? Because the calcium needs to bind to troponin so that the myosin binding sites on actin are actually exposed, right? Because that'll push tropomyosin off of the myosin binding sites on all the different globular actins. So you need calcium inside of the muscle fiber in order for it to contract. And you also need plenty of ATP, right? There's always some ATP being produced in the muscle fiber and in every cell in your body, through some of the pathways that we discussed at the beginning of the semester. But you can probably imagine, as you ask the muscle fiber to contract more forcefully and more frequently, it's going to start consuming more ATP, right? Because every single time the myosin head cycles, what does it do? It consumes ATP. So if you ask a muscle fiber to contract a lot, it consumes a lot more ATP because the myosin heads are cycling way more. And then what does the muscle fiber have to do? It has to start making a whole bunch more ATP. It has to meet its ATP demand. Anyways, the point is, if you want a muscle fiber to contract, you need ATP within it, which we can assume to some degree is already there. And you also need calcium within the fiber, which is not there initially. Okay. If you just looked at a resting skeletal muscle fiber, there'd be very, very little calcium inside of it. And if you want that resting skeletal muscle fiber to now contract, meaning you have myosin and actin interacting and force to be produced, then you somehow need calcium levels to elevate inside of the fiber. And that is done through neurons, which leads back into a lot of the conversations that we've already had in this semester. And they just keep coming up, and they're going to keep coming up for the remainder of the semester. Okay? Muscle fibers can generate action potentials just like neurons can. And neurons and muscle fibers are actually going to interact with each other through action potentials to influence the amount of calcium in the fiber to allow it to contract. So we can acknowledge for a muscle fiber to contract, we need calcium inside and we need ATP inside. We know where the ATP comes from. We know that there's some ATP in the fiber. We don't really know where the calcium comes from yet. And additionally, there's no just natural calcium floating around in the fiber. So that calcium has to be released somehow, and it's going to be released through action potentials, as you're going to see in the coming lectures. So the goal, of course, is by the end of this for you to be able to see the entirety by which the nervous system allows muscle fibers to contract.